Looks like it will be only our department today.
Well, there are some people outside the department also.
Oh, there you are. Yes. Since it was a holiday today, I was not
Thanks,
sure.
Gagan.
Well, Kajuri is there, and that Gautam. I mean, there are a few others. So anyway, I'm not the only one.
Okay, we are live now. So I welcome all of you for this session of the Wednesday colloquium. And it's my pleasure to have my departmental colleague, Professor Sudipta Maiti, giving the Wednesday colloquium today. Before I introduce formally Professor Sudipta Maiti for everyone here, I would like to tell and remind for those who are joining for the first time that the Wednesday colloquium has been a traditional forum for all sorts of scientific communication and discussion in the institute for as long as the institute has been around in Mumbai. It was started by Professor Homi Bhabha, the founding director of the institute, and it has been one of the major activities of all faculty members getting together in one room, usually when the meetings are physical, and all the faculties means scientists from physics, chemistry, and biology background. So just to have a discussion from 4 to 5:30 on a Wednesday afternoon. This particular session is no different, where one of our leading experts in biophysics, absolutely frontline in the world, Professor Sudipta Maiti, is going to talk about some of the very exciting work that's going on in his lab at TIFR. So Professor Sudipta Maiti actually, to all of us, is very well known in his background as a physical chemist, spectroscopist, and a biophysicist, getting well trained from the labs of Professor Robin Hochstrasser for his PhD and Professor Watt Webb from Cornell for his postdoc. His group has pioneered use of optical tools, both microscopy tools as well as spectroscopy tools, to look at very difficult problems in biology. Being a physicist, his perspective matters for all biologists, because he actually tries to understand the mechanistic views of different kinds of biological phenomena, and especially the disease causing amyloids that he essentially works on. Today, he's going to talk about amphipathic molecules in biology. Sudipta, without further ado, you can please proceed with your colloquium.
Thank you, Jyotishman, for that bit of undeserved praise. It's great to be back as a colloquium speaker after a while. It's also sad. I'm really yearning to sit and listen to people or talk in AG 66. We really miss it, I think, every one of us does. All right, that has to be a very happy new year to many of you, plus minus 1 day, I guess, for many of us. And it's not so happy in Bombay because we're just starting a lockdown a couple of hours after my lecture finishes.
Sorry,
just
interrupting. Sorry, Sudipta, just interrupting. I just want to remind the audience that you were also a host of the Wednesday colloquium, and you have a connection to this series. So thank you for doing that as well.
Most welcome. And welcome to this Wednesday colloquium. So today I'm going to talk about amphipathic molecules. For those of you who do not know what an amphipathic molecule is, it's simple. It's a molecule which has parts which love water and has parts which hate water. Okay. Of course, those are very colloquial terms, and I can explain it later if you want. But that sort of would do it. So here in the picture that you have in front of you, you'll see a few things such as this dark molecule. It's called serotonin. It's one of the two players today that I'll talk about. You see it's hanging around something which actually is a fat bilayer. I'll talk about that also. And it has parts which are polar, so which is water loving. It has parts which are non-polar. So it really is a bit confused, or on the other hand, you can say it's opportunistic, because as we know, life is mainly the first, if I have to make the first division, it's water, the aqueous phase, and the non-aqueous phase. There are lots of molecules, probably the majority in the aqueous phase, but there is also a very substantial chunk in the non-aqueous phase. And this is absolutely needed for life, as I've come to it. But what we have been working on, especially a new type over the last 18 months, maybe 2 years now, are molecules that were not really appreciated as amphipathic and known to be working through the aqueous medium. But suddenly we find that they're amphipathic and they actually are doing something in the other phase. Okay, so it's about molecules which either you can call them confused or you can call them opportunistic. And that's an interesting place to be in. I mean, it's not black and white. They are all very gray molecules. So before I get to the topic of the colloquium, let me quickly run through the stuff that goes on in my lab. Of course, it has been a while since I've given a colloquium, so there are many, many stories that has come up. Some are matured, some we're working on, some new stuff has started. And some of these I would not be able to talk about. So let me quickly touch upon these. This page that you see, 4 things, so this is about amyloid aggregation. Nothing to do with membranes in solution, which I will touch upon a little bit, but mostly done. And this is something that we'll touch upon. One of those 2 molecules I talk about is an amyloid molecule in membrane. And this is something that's same amyloids in neurons, very biological stuff. I would not touch it. But the most interesting stuff that's going on, the newest results in the lab are on these. And I think we have some very significant results, but they're not mature enough yet to talk about. There is something else very, very cool about modulating protein folding using something we call a xenonucleus. And so this is about designing stuff to fool proteins. Again, I won't talk about it, but interesting stuff. Sorry. This is about imaging neurons. We started this lab, well, in a lab form, since it took a couple of years to get even lab space in about 2001 to get space and a laser, which is 3 years after I joined. And this is stuff that we started at that time. There have been periodic, and this has been, I won't say the mainstay, but there is one sort of side stream always of this imaging neurons and coming up with new ways to look at neurons that other people cannot look at. And we have periodically students say, hey, I know that you are known for this. Can you do something about it? There's one such student, Dev Shankar, right now. And hopefully we'll do something new with this. Okay, so then developing optical techniques other than this neuronal imaging. Here we also, of course, interface with commercial vendors. We have commercialized this instrument from TIFR. You can see it, and we keep developing new and new techniques. This is related, mostly related to the question that we ask. I think where our strength lies is in actually inventing or developing or at least modifying the instruments that are there, methodology that is there, and take that advantage. If you don't do that, you have to be really, really creative. So in my limited brain, I just go for developing new techniques and see if I can get new answers. This is something that was spawned by in last April, when everything closed down, and we are only allowed to work if you're working on, say, COVID-related stuff. And we did do some COVID-related stuff. Much of this has remained a promise that we haven't gone to, followed through with. There are some difficulties. It has been fun. Why it has been so difficult to solve that? Because it has been difficult for many people to find a molecule that will bind to the COVID protein and stop the infection. It's not very easy. You can get an antibody, but you can't get a small molecule. So that's another story that's developing with many of my colleagues. There's been a wonderful collaboration because we just called each other and say, what can we do about COVID? And there are lots of hands that are up, including many from my department, many from this campus, but also from all across TIFR and outside. This is something I'm going to talk about. This is the second type of molecule, neurotransmitters. I'll talk about their membrane interaction. This is the stuff that we have been doing for the last 2 years or so, which was quite unexpected. We're seeing some fun effects, ways that nature cells talk to each other that are not quite well appreciated. Okay, so let me start. So the 4 things that I'll tell you about today, let me set my clock also. Okay. 4 things that I'll tell you about today. The first thing is that amphipathic molecules and why are they important. It's fun to be working with them. They comprise of things such as membranes, some proteins, actually most proteins, as I'll tell you, though overall the proteins are mostly hydrophilic. I'll tell you about that. Some small molecules. Okay. And I'll say that this amphipathic membrane actually makes a cover to the cell, but the cell needs a window to the outside world. The cell needs to be disconnected from the outside world so that it can maintain a non-equilibrium local region, which we call life, but also needs a window because it needs matter and energy both to take it in and throw it out, and signaling, of course. Matter and energy is just low-level stuff. The signals are the main thing. And so therefore, it needs an outside world while it needs to have a boundary. So that's kind of a dichotomy that life has solved in many beautiful ways. So we'll talk a little bit about those. Because what I'm going to talk about are these two molecules. They're masquerading, I think, as some of the well-known molecules which are needed to provide that viewpoint, to window to the cell. And they sort of masquerade as that. And some of them are toxic proteins causing things as Alzheimer's, Parkinson's, Lou Gehrig's disease, Stephen Hawking's disease, ALS, and all that. Some are transporters, some are just transponders. And finally, I'll come to so this is the first type of molecule. I'll come to the second type of molecule. Signaling molecules can the signal directly to the membrane. So the amphipathic small molecules, which I showed in the first picture, they can talk to the membrane. That's what we find. So, okay, let me start by saying life needs amphipathic. Lot of people, lot of very smart people work on creating life in the lab. Okay, so that's been going on for a while, and every couple of years there is something that tells us that they're getting closer. In fact, one of my lab mates, is very well known now, Professor Petrashvili, my lab mate in my postdoc years, proclaimed 2 years ago that in 10 years she is going to create life in the lab. And I sort of believe it. May not be 10, it may be 20, but we're not very far. But what is the challenge? The challenge is that you have life. If you look at this sort of spherical stuff, you have life which needs to have some molecule, some chemistry going on in a small region, which is aqueous. All of those molecules are very soluble. Some are very charged. This blue thing, I would say, is an RNA, which is the precursor life molecule. Even before proteins and even before DNA, we had RNA. And most likely the initial life was RNA-based. RNAs masqueraded as enzymes. RNAs, of course, carried the genetic information. And this is something that has evolved, but you will think if it has evolved, this is something in the past. Think about SARS-CoV-2. SARS-CoV-2 is an RNA virus. It's not a real living thing in some sense, but it's an RNA virus. So the designs have stuck around. But if you have to do chemistry, typically if you ask Jyotishman, he'll grab a beaker and try doing chemistry because you don't want your whole of all the chemical entities to go out, either evaporate or mix in the solution and go to the drain. You have to have a boundary. You hold the chemistry in some place and then put in something, and that does something interesting because it's not at equilibrium. It gets photoexcited, it gets thermally excited, whatever it is, and it does something. To do that, you need to have a boundary. And this is the boundary that the people who are trying to create life in the lab are trying to create. And we think that this boundary got created with amphipathic molecules. Molecules which we now call lipids, and there's a cartoon of it. Of course, the yellow dot and two tails. The yellow dot is something that is polar, could be charged, could be uncharged, but polar, water-loving, hydrophilic. And these two tails are hydrophobic. So if you put a lot of these molecules together, they self-organize. I mean, that's of course the challenge for creating life. It has to self-organize. You cannot just build it piece by piece and say, oh, look, there is something that's living. The life has to be self-organizing. So it self-organizes because, of course, the hydrophobic parts, all these brown tails, actually try to hide from water. That's just going down the free energy landscape. And in a sense, in a way that it organizes, the yellow hydrophilic parts face the water. Water is outside. Sorry, it's black and blue. I don't know how that came about, but it's all blue, both sides. Water outside and this is water inside. Water inside then catches the important molecules. Don't let them go out. While energy, other molecules, building blocks have to be brought in. I'll talk about that. But now you can do some chemistry where everything doesn't disappear. I mean, it takes enormous, probably took at least 500 million years to come up with a configuration like this, which then propagated and made life. So you don't want to waste those. So talking about the first successful ancestor, you see the last universal common ancestor. There is a creature called Luca, okay, in evolution. And of course, this is from some metaphysics things I found in Google. It's not very metaphysical. Of course, it doesn't have eyes, but it probably looked something like this, just very simple cells. But it's the last universal common ancestor. That part is scientific. So if you look at the genetic evolution of things, it points towards one cell actually having spawned this. That doesn't mean that was the first living cell or first living thing. There are many, many cells, maybe millions of cells were there, but finally, one guy succeeded in spawning things which stuck around till today. Okay. It's
So
just like the human race. You know, there's Lucy. So this is Luca for the single cell, and Lucy for the lady that we think all of us are our mother, the original mother. Because if you look at the mitochondrial genome, which gets passed on from ladies to ladies, the all mitochondrial genome has a very convergent, backward convergent point, which about 100,000 or so years ago seems to have come from a single individual. So in that way, we are all brothers and sisters.
There is a question by Gautam Mondal.
Yeah, sure. Gautam, go ahead.
Okay, I don't know how you knew that, but I didn't. Yes.
Yeah.
So I mean, this first cell, you know, so there's this theory by Freeman Dyson, who is not a biologist, called the origins of life. So they talks about two origins of life, and the protein life, according to this theory, protein life formed first, which could do only metabolism and stuff and couldn't replicate. And then DNA, RNA life happened later on. So what's the idea of this Luca thing? I mean, is it, what could it do?
That's the first one that could actually store information and procreate in a way that the information got transferred to its progeny. Okay. So what is life is itself is a slightly complicated question. In fact, if you look back and look at the person who, Craig Venter, who was the original person who was doing the Human Genome Project, he claims to have already made life in the lab. But if you look at it, then it's not something that is procreating, maintaining its lineage through procreation and through information storage. So as far as we know, and for reasons that are very biophysically obvious, proteins are not good information storage things. They cannot replicate. They cannot make copies of themselves by themselves. Yes. So it has always been like this, that you have something that carries the information. It creates the protein. The proteins, after all, are the functional elements. DNA and RNA, of course, RNA in ribosomes and all do carry out things which are supposed to be the historical, this evolutionary thing. So in the sense that you have molecules carrying genetic information, able to replicate that information in a molecular form, this is the last universal common ancestor. The thing comes from, actually, if you look at the three trees of life, the animal kingdom, plant kingdom, and the archaea, you see there are some common things, and which convergent evolution can only be around about 3.5 billion years ago, which is possibly when the first life emerged, a little, maybe a few hundred million years after the first life emerged. So we do have a common ancestor in that sense, and this is possibly, there's all a theory, of course. It's sort of a prediction looking at the evolution back. The proof is very tough, but yeah, in that sense.
Thank you.
Okay, Sudipta, just on a lighter note, your third reference, Joshua, hey, what is this a scientist?
I do not know. It may be a scientific cartoonist or somebody, but it's his copyright, so I wanted to put that. I'm not laughing, okay? So don't laugh. All right. So amphipathic molecules. So life needed a membrane, and this is something that is very hard to create for people who are trying to create life in the lab. And now there are two theories have emerged. One is this prebiotic pool. You know, you had lightning, whatever, the Miller experiment, etc. But there is a competing theory now that it actually came out deep inside the sea in the thermal vents, which have very high energy. You have sulfur, hydrogen, carbon, etc., and then life. So those people, especially those people who are talking about these deep hydrothermal vents, greater than 100 degrees Celsius temperature, of course, it's so high pressure, it's all liquid, and they have very hard time making a membrane. So the latest papers, if you look at from that camp, are making membranes, making these boundaries with special types of lipids which they say don't exist anymore. Our types of lipids, so these are lipid molecules. Sorry, I'm getting ahead of myself. So I told you that there is a head group, hydrophilic, and the tail group, which is hydrophobic. If you look at a ball and stick kind of model, it looks like that. This is polar, so it's something like this blue dot, and this tail, these two tails. Okay, and this forms the boundary of the cell. So that everything can be contained. And okay, sorry, where did everything can be contained, and this is obviously very nicely self-organizing. You can create in the lab all the time. My students do it all the time. You put some lipid in water, sonicate it, you'll have vesicles. Okay, vesicles are these balls, and you can trap things inside those vesicles. And the last bit that I can tell you is that two of the vaccines which are there in the developed world are this vesicle carrying RNA, very similar to your cartoon of the first life form. Okay, and that's why I think some people are scared that they are going to get mutated or something. I know some my friends are scared and not taking the vaccine. But yes, so you need a boundary. But it's not just these lipid molecules which are so easy to self-organize. Proteins, large proteins, are actually amphipathic. So proteins are these long chain molecules which have hydrophobic and hydrophilic parts. I'm sorry, at this rate I'll be at 2.5 hours. So hydrophobic and hydrophilic parts, and it has to wrap itself around and hide its hydrophobic parts so that only the hydrophilic exterior is there. So it's not so simple, because this, in the long chain, if you just pull it apart and make a single chain, the hydrophilic parts are here and there. Okay, you know, there are 50 hydrophilic parts at different places, 50 hydrophobic parts or larger, whatever. And then you have to compress this chain and have only the hydrophilic parts showing outside. They are not continuous. So this is a big problem. It's well recognized now that the proteins have evolved not just to create the function or give the living advantage for doing the function or being a better enzyme. It's also equally well selected for folding right. So foldability is a huge problem that nature had to solve during protein evolution. So I'm showing you something called myoglobin. It's a protein. And you see the blue are the hydrophilic parts, yellow are the hydrophobic parts. When it's folded, mostly it's blue outside, but I agree there are a lot of yellow that is showing. But if you take a cut through the middle, you see that the blue is really outside and the middle is almost entirely yellow, hydrophobic. So it's also an amphipathic molecule, but it has self-folded. Okay, the lipid molecules, you remember, they need a lot of lipid molecules to make a structure that will be stable in water. Here, each individual molecule self-folds, and this is what primarily drives protein folding. Other energetic contributions are lower in energy, typically free energy. And you create a molecule which is ultimately hydrophilic, because as long as the surface is blue, water loves it, and the protein can be soluble in water. Okay, but some small molecules, this is about a protein, can also be amphiphilic. Okay, here is cholesterol that people of my age group and older all worry about sometime or the other. Cholesterol is something that is actually somewhat amphiphilic. It's not very soluble in water, but it does have a polar group attached to it. So it just doesn't go into the fat part. So this is again the membrane. Blue part is the hydrophilic, the head groups of the membrane, the tails here, and the yellow things are cholesterol. So they go insert into the membrane and do something very useful. They help the membrane organize in different ways. They maintain the stiffness of the membrane, and that's very important for our brain to function. In our brains, molecule per molecule, there are 50% cholesterol. So that's why actually pregnant moms are specifically told not to diet, not to go on diet. We need cholesterol, absolutely. Okay, the stupid heart thing, gets clogged occasionally with cholesterol, but the brain needs it. Okay, so another molecule that I'll talk about is sort of in this vein, small molecule serotonin, which is known as a signaling molecule in the brain, that were not appreciated as an amphipathic molecule because all its function was supposed to be in the aqueous phase. That we found, like cholesterol, inserts in the membrane and does something quite fun. So I'll come to these two molecules. So part 2 of my talk. The cell needs a window. Okay, so now we have a membrane, and suppose my friends can create a membrane in which they have a machinery which can utilize energy and molecules from outside and do something. Ultimately, they need to bring in those things. As I said, energy and building matter has to come in from the outside so that you can build life's molecules. But also signals. That's very, very important. When to run, where the food is, etc. Even a bacteria knows, and it has to have molecules that can let it communicate across the membrane. The moment you close it off, it becomes me or a living thing. But the outside is extremely important to communicate with. There are many different types of molecules, but the 3 major things that lets the cell communicate with the outside world are transporters. So they have sort of little regulated holes in the membrane. They're proteins, and they're extremely well designed so that they can sit in the membrane. Part of it sits in the aqueous phase, and they can bring in stuff very selectively. You'll be surprised that the brain won't function, and you talk about sometimes sodium potassium imbalance on all that. Brain won't function unless you had ion transporters, which can actually transport a bigger ion and stop a smaller ion of the same kind. You transport potassium, not sodium, or transport sodium and not potassium. And so transporters are there, which are important, transport ions and other molecules, food of some kind or the other. Receptors are there, which is for signaling. You don't need to open a door all the time or open a window all the time. All you need to see is look who is outside and understand what's going to happen. So these are receptor molecules. There is no physical breakage of the membrane or physical hole in the membrane. They are transmembrane. They will sense something outside and tell inside what's happening. And most of the modern drug molecules are developed for either the transporters or the receptors. Okay, especially things which are neuroactive molecules are all working. Everybody is working on these, either of these things. Then there is a bigger thing, when you can't do it by taking single molecule by single molecule or just sensing things, you want to throw out a whole chunk of things outside or grab a whole chunk of thing inside. Okay, so your antibodies, etc., which are now hopefully those of you who are vaccinated, being generated. Ultimately, there will be T cells and B cells which have the ability to engulf a cell which is infected, and that they have memory of, let's say, SARS-CoV-2 or something. That's what they do it by endocytosis or some different types of endocytosis. They will call it phagocytosis, but I'm using endocytosis in a very loose way. But the process by which it engulfs something and takes it in, or exocytosis is just the opposite. It opens up and throws something out. So these are the 3 things, and they will play parts in this story. Okay. So let's first talk about transporters a little bit. Transporters are, as I said, molecules that sit across this membrane, cell membrane. This is a transporter. Sodium ion comes in, goes out. It only will, there's a rectifier, so it will only transport sodium one way. And similarly, there are other transporters. There is a bigger transporter, can take in bigger molecules. These are called porins. Beautiful structure. You see these golden things. You see these little tapes. Those tapes actually are atomically accurate drawings. Okay, it's tough to appreciate that. Those are backbones of molecules whose crystal structure have been solved. They're not just cartoons. They are cartoons, yes. Protein doesn't look like that. That the tape and the arrow in the tape has a meaning, but believe me, they're drawn from cartoons drawn from atomic coordinates. Porins are of a family of different kinds of pores that they can open, and I'll come to porins because they will be very important there in the next thing that I'm going to talk about. Pores are, as long as they're regulated and well designed, well regulated, they are good for you because it helps you take in something and throw out something. But if an external molecule forms a pore in a membrane, that cell is probably going to die. So that's a. So this is how porins look like. It's an amphipathic molecule. How do I know that? So this is the top view. So the membrane on the left side, the membrane is on the plane of my screen, and these are the holes through which you can go right in to the cell. Okay, there's a trimer, triple hole. Okay, but the red part and the green parts have connotations. I have colored them from PDB, the protein database, and you can the red and yellow parts are hydrophilic, and the green parts are hydrophobic. So you have to imagine that sitting in the membrane is about this thick from here to there. Okay, and it's all around. So this is a barrel sitting in the membrane, and all this part is facing the lipid tails, which are hydrophobic. But this part and this part is facing, let's say, the outside, which is aqueous. This part is facing the inside of the cell, which is also aqueous. So you see a gradation of red and orange here, and very little in this plane and the middle plane, and then a lot of red and orange here. This is how an amphipathic molecule exquisitely self-organizes. And the fact that such a molecular machine can just by free energy organize itself in the membrane is just astounding. And but it does it all the time. This is what life is all about. This is a ribbon drawing of the same molecule, microbial porin, and you see how nicely the actual backbone is organized with some little loops, disordered loops. Okay, receptors are similar. They, this is a receptor and transporter together. This is serotonin.
Vivek has a question. Vivek has a question.
So what is the diameter of that pores, Sudipta, that you showed from the top?
Yeah. So porins are funny because depending on the number of strands that you have, it can increase in diameter from 3 to about 20 angstrom. So it can be very large. You see, there are some porins like this one, Amphipathic. Okay, so that's very large. Nature has hit upon this design and she's going to use it. So when she needs a bigger pore, she puts, I'm talking in a funny sense, it's encoded. There are about 24 strands. It will make a big pore, about 2 nanometers or so, but it can be as small as 6 strands, and it can be a few angstroms.
Okay.
So these are receptors which senses molecule outside and does something inside. And to hurry up a little bit, I see half my time is gone. Okay, and this is exo and endocytosis. So the cell has something inside. It wants these molecules, little blue molecules, to go outside. Okay, these are actually signaling molecules. This is how your brain thinks and feels emotion, etc. They have to talk to each other. These are the messenger molecules, and this is one of the processes. And they then go. This is also a lipid bilayer membrane. This is also a lipid bilayer membrane, and they have to fuse together. This is the dance that our Devashish, who is in this audience, is a master of, and they finally fuse, open up so that these things can go out. Okay, they can be very different molecules. And this is a process which is the cartoon representation of that. It shows you that it has to bend. There's a high free energy thing. There's 10 kT roughly. It has to overcome for this hole to open up. And many biophysicists work on why this happens, how this happens. There are some of the molecules that make it possible, etc. But the bending, ability to bend, is very important. So the membrane should have an ability to bend. Of course, it's quite floppy, it bends, but that rigidity, change of rigidity, is something that can completely change the picture for exocytosis. It can make, if the membrane becomes harder to bend, it may make exocytosis or endocytosis difficult. It becomes easier. It should be easier to do exocytosis and endocytosis. And I'll talk about that. We're doing some wonderful stuff with Devashish, so we won't be able to talk about that, but this is a different context. So let me just quickly run through. I have hopefully about half an hour. So these are the two kinds of molecules which are, I say, intruders. So till now, I talked about things which life uses. Okay, they're very useful molecules. You do need hydrophilic stuff. You do need hydrophobic stuff. You do need this amphipathic stuff which go across the membrane. But there are proteins and there are small molecules which mimic other molecules which we know are useful for nature. One certainly is not useful. In fact, it's very toxic. There's an amyloid beta. This belongs to a class of molecules which cause these incurable diseases, as I said, from Parkinson's, Alzheimer's, and other things. And this neurotransmitter molecules, which the cell throws out by exocytosis. They also seem to have membrane affinity for a reason that nobody understands, but I think we are beginning to understand it a little bit the last 18 months or so. So let me come to the first class, the amyloid proteins. And after 100 years of Alzheimer's describing the disease, and over 30 years of intense research, we don't even understand why this Alzheimer's disease happens. Okay, that's a shame, but we don't know the mechanism. There are speculations that it happens. The closest that you have got to is possibly it happens because the protein molecule that causes Alzheimer's is a small molecule. And possibly somehow it's aggregating and forming a porin-like structure in the membrane. It's a little bit of proof with a lot of imagination thrown in, but we started on these games trying to understand it over 8 years ago now. And I think we have gone somewhere which lends some little bit more firm ground to that hypothesis. So we're still far from understanding and solving it fully. So here is the protein, amyloid beta, and it misfolds. It comes together and aggregates with others, forms these multimolecular bodies. They're called, and the very mature bodies are called amyloid fibrils. These little things I call oligomers. Oligo means few, maybe 3, 4, 5, 6, and these are monomers. There's a scale difference here. Of course, this is a single molecule, and that is, in this scale, is one little thing here. So as you go from left to right, the scale is sort of zooming out. Okay, but finally, what it does is extremely, it's a human tragedy. The brain gets eaten up sort of by proteins, not by bacteria or viruses, by proteins, and the brain shrinks and lots of neurons die. And ultimately, you forget who you are. It affects the memory first and then affects many other things. So it's a huge burden, and it's not a very uncommon disease. All of us would probably know somebody who would have this disease or one of these class of diseases, as I said, Parkinson's and others. Okay. I definitely have things in the family. So anyway, so it's just, as I said, it's a shame that we don't understand how this happens, but perhaps it happens because the amyloid beta molecules is a small molecule. So instead of having these 50 different parts which are hydrophilic, which it can have ways to combine and hold together so that it's all hydrophilic outside, all hydrophobic inside, it can get together with other copies of itself and make up for the lack of length. Because when a protein, I talk about length, we typically talk in terms of amino acids, and even smallish protein typically has a few hundred, and this guy only has 40 or 42. There are slight variations. And so there's no way it's going to be able to fold together very well. So it has to be, so it has to get together with each other. So a lot of hydrophobic parts are exposed. So like the membrane self-organizes because hydrophobic parts come together, they can come together and make some structure. And that kind of structure can sit in the membrane. I mean, I'm showing a helical structure. It need not be helix. A porine is not a helix, it's a beta barrel, but it can sit in the membrane because part of the structure will still have hydrophobic residues exposed, part will have hopefully hydrophilic things, and then it can sit in the membrane and can make something. And this is our best guess till now, that these molecules are becoming toxic and killing the brains because they are making unregulated porin-like channels. Okay, as I showed, as I said, that neurons absolutely depend on exact regulation of, let's say, sodium, potassium, whatever you have. And then if you punch a few of these holes in the membrane, it's just not going to be able to do its job, and it's going to ultimately die. So that's the best theory. And we set out about 6-7 years ago to see whether that theory holds any water. There are very good reasons 7 years ago that actually it's all nonsense, okay? And nobody had an answer, so we wanted to ask that. The first thing is that, can the oligomers really attach to the membrane? How they sit in the membrane is very difficult to know and sit, because you have to develop methodologies for it, few of which we have developed. But if you look at this graph, I'm just showing attachment. If you leave them in water, they're amphipathic. Some part will remain in water, a fraction will go and attach to a membrane. So we do vesicles, put vesicles in water is there, and we see what is attaching. And there are techniques which these graphs comes from. They're called fluorescence correlation spectroscopy, which actually gives you the size, the diffusivity of molecules, how quickly something diffuses, that relates to the size. And we know when we are dealing with monomers, we know when we are dealing with oligomers, and you can tell when you are dealing with monomers, which are fluorescently labeled, that's the only way you see the molecules, are attached to a bigger object because then they will diffuse slower. So this curve going to the right is actually, so black, blue and green. Black is sort of monomers, blue is sort of oligomers, and green is something oligomers attached to vesicles. Okay, because vesicles are much bigger body. You see, this is a log time scale, so the difference is quite large. So what we realized about 8 years ago is that oligomers can attach to the membrane and monomers cannot, which is at once explains that why every one of us has the monomers, and only some unfortunate ones have a large portion of oligomers. Why it may be toxic. It doesn't prove anything. It just shows that it has a different property. It can attack at least attach to the membrane. Whether it can attack the membrane is a different question. It can attach to the membrane. That's a measurement that we found ways to do. And but are they ion channels? Fine, they're going attaching to the membrane. They will be doing nothing. Some people showed that there is ion poration, so ion can transport across. They wanted to know structurally, is that possible? Or are they looking at some other, like a detergent-like effect? Okay. So what you asked is that, do they have a particular structure? If you have an ion channel, you saw those ion channels. The first guy who solved an ion channel knew that he was going to get a Nobel Prize. It was so important. And 1998, Roderick McKinnon solved the ion channel, first ion channel, the potassium channel. In 5 years, he got the Nobel Prize, and it was guaranteed.
What's the question?
It's an exquisite,
Yes,
complicated structure.
yes. Please ask. Ullas has a question, Sudipta.
Yeah, Ullas. Right.
Sudipta, if you can go back, please.
Yeah.
Yeah. So all of this is also concentration dependent and possibly a nucleation event involved. Is it possible to actually see what's the nucleation, both in terms of where in the protein and the concentration dependence?
Yeah, so that's a problem that we separately worked on a lot. I'm glad you asked the question. The thermodynamics of aggregation, and our current colleague Kanchan Gadai, when he was a student with me, did a lot of very good stuff with that. So I'm not going to talk about that, but these oligomers most likely are pre-nuclear stuff, okay? And they exist in a quasi-stable way. That's why they're very hard to actually characterize. When the monomers start growing, you have a supersaturation, you'll have some stability or quasi-stability to these oligomers. They are dimers, trimers, tetramers, etc. When the number becomes a certain mar, I think under certain condition, Kanchan's experiment said that number is about 20. Above that, the free energy curve turns around and you have nucleation-dependent growth. Yes, but nucleation is something that we actually established. Starting from the fact that these guys have a well-defined saturation concentration. That's my first paper many, many years ago in this field. But it was fun working out the thermodynamics.
Yeah, so
talk about
that's just the first part of the question. No, that's just for the first part of the question. But the thing is, in reality, what is also happening is these proteins are not present alone, and also your membrane composition also might vary a lot. So the entire thermodynamics of nucleation and let's say spreading of this phase transition, if I can use that word very loosely, how much of it is dependent on the other interactions and the membrane interactions?
Very good question. Actually, I mean, these are the questions that everybody should have asked even 20 years ago when some of us started asking the question. If you think of nucleation and aggregation in solution, and you say that stuff goes and sits in the membrane, I'll say that's bunkum. That's absolute nonsense, because it aggregated in solution because it was doing it to hide its hydrophobic areas. Now you are saying that you see some structure in solution, and that same thing will happen in the membrane. That doesn't make any sense at all. Say modulated, highly modulated by the membrane, and a membrane-mediated nucleation versus something that nucleates in the solution and goes to the membrane are two separate processes. But what we have found out is that the oligomers are made in a way they can be, that oligomers I mean n less than 10, okay? Made in a way they can be actually partially stable in aqueous phase. And of course they change their conformation, but they can be there also in the membrane phase, okay? So it can form outside and go there, but also separately aggregates can form in the membrane. Both are possible, but I think as of now, the oligomers which are forming outside and attacking the membrane are perhaps one of the major causes of the disease. But yeah. Okay, so a small oligomers acting like porins. Quickly, I'll run you through the questions. porins are antiparallel. Here it's technical word, but you see that's how these things are, this green strand is going, there's an arrow here. Okay, then it turns. It's the same molecular chain, and it comes here, and the arrow is on this side. So arrow up, arrow down, and that's called an antiparallel beta sheet. Okay, this is all porins are like that, all porins. Okay, antiparallel beta sheets, and this itself was an unanswered question, elephant in the room, because all the question that Ullas asked, all the solution state structures of aggregates talked about parallel beta sheet. You might think that's a small difference. It's not. It's a completely organization of stuff. But we found out actually antiparallel beta sheets are there for the solution state oligomers also, which is different from the parallel beta sheets that larger aggregates have, and they can sit in the membrane. But let me get back to the story. So it has to be antiparallel. It has to have a specific orientation. It cannot be like horizontal if the membrane is in the horizontal plane and all angles, because if it's an ion channel that something is making, it better have a proper shape, orientation, specific number of strands, and specific placement in the membrane. Okay, so these are the sort of criteria we put about 5 years ago, saying that if you're talking that these guys can form a self-organized ion channel, and I have to call it an ion channel, it has to satisfy these at least. And then, of course, it has to be functional. So we went one by one and looked at this. I will skip through these. We did find there is a conformational change between monomers and oligomers, but this is old data. We found out with this some structural change that you could characterize very much that monomers and oligomers are different. And then we were talking about nanometer scale things when Mr. Madhu came along and said, well, I can give you angstrom level stuff. Then we had a lot of fun together developing new techniques with solid-state NMR looking at oligomers. And we finally had some atomic level information about this oligomer. As I said, the structures are very difficult to characterize. This is all Madhu's data in between. This is all our data, but they're all the same sample. That's the fun part of it. We made a technique where two things could come together and we could do it. And this is why I love being in TIFR.
Anup has a question. Anup has a question.
Anup has a question.
Anup, can you ask?
Anup? Oh, no. There was not a question. I accidentally unmuted.
Okay, no issues.
Oh, okay, all right. Okay. And we found out some atomic level contacts. This is F is a name called phenylalanine, L is a name called leucine. They're names of amino acids, and 19 is the number. These proteins are sequenced from so-called N terminal, the amino terminal, to the carboxy terminal. So 1, 2, 3, 4, 19th residue and 34th residue. Told you this is about 40 residue stuff. So there is an F19L34 cross-peak, means two things which are not next to each other, they have to fold and come together. So all this we found out quite a few years ago. The question we are thinking is that we are biophysicists and we are stressing the structure so much. Does the structure have something to do with the properties? And we always tend to believe it's like an axiom almost, but is that axiom true? One needs to question oneself. And we found out that that little contact, we said, and it goes very well into the membrane, and I showed you the monomer versus oligomer. Let's change that little contact a little bit. And proteins come in these two flavors in nature for reasons unknown. Nature has built up from the L type of amino acids. They're isomer and of opposite chirality. And you have the D types, which you can synthesize and you can make proteins out of those. So we took one. So the L will, it's a very crude way of saying it. L goes this way, then the D will go that way, okay, in a chain, along a chain, if you hang them. So this is the L type, and this is the F19L34 residues contacting each other. I'm not showing the other contacts, just these two. And so this is like a zipper. And then I replace this phenylalanine with a D type. So it hangs on the other side. So the zipper opens. Okay, very small difference. There is no chemical difference at all, other than this handedness. Okay, the charge, the hydrophobicity, whatever have you, they remain exactly the same. And this completely changes its ability to attach to the membrane. So this was beautiful. This is really one of those things that you live for. And you found that, yes, it completely changes. One single L2D mutation. So a lot of work done later on, some with us, some by themselves, with Daniel Huster, who was a visitor for 6 months last year. And he has done some much more beautiful work, making little changes here and there, and showing that not only attachment, but cell viability changes. This part, these two, of course, we are part of. And the main thing is that little change in structure completely can change this amphipathic, not the amphipathic nature so much, but the ability to organize in the membrane. Okay, so the structure is important. So why is the structure important? Yes. Oh, Alok. Yeah. How? Hi, Alok. Go ahead, please.
How did you manage to attach this single molecule outside versus inside?
Okay. So all you need to do is you have to synthesize the protein yourself. And I said, if you give it to nature, nature will always all do L, except for a few. Gramicetine is one. But it will do all L. So we synthesize the protein ourselves. So one by one, we couple. And we're extremely poor at it because to do 40 residues will take me 3 days or something. Newer machines can do it in 3 hours. But it gives you the ability to put a non-natural amino acid. So this D thing, D-phenylalanine, is a non-natural amino acid. In the sequence I built from this side, I come in, and here in the tube, which is a reaction vessel, I put in only D-phenylalanine. It doesn't exist in nature, but you can make it. You can buy it. And it incorporates the D thing and keeps going. Okay, some beautiful work with D-dependent peptides, which can be very promising medicines, are being done by Kullaneshwar Mondal in TIFR Hyderabad. But there's just one small. So it's, yeah, artificial.
Sudipta, I have one
Vivek.
About the solution NMR. So when so is it not possible that when I do a freeze drying to solidify your protein and then do the NMR versus protein in the solution? So how do you test that there is no change during the sample preparation?
Very good question. So we can test not at the atomic level, but you remember that we first came to it using FRET, okay, and FCS. So those are fluorescent labels attached. So they'll give you nanometric, sort of little sub-nanometric information. So what we did is that in the solution, we do it because you can do it in solution. We made the powder, we did the solid state stuff, and it had a little bit of that in the label stuff. We came back, redissolved it, and asked the state stuff. FRET. Okay, and we did the FRET, and it remained the same. So that's there in the paper. It's exactly the same question. Very good question that the reviewer asked also. And we are also curious. So we had that answer. Okay, so so we had to find out some new techniques to find the structure in the membrane. As remember Ullas's question, I mean, so what is some structure you know in the solution? How does it matter? Membrane is a completely different ballgame. Absolutely. So we didn't know what we'll find in the membrane, and it was much harder to do similar stuff in the membrane. So we could do a little bit, and we had to come up with a new technique, which is plasmonics, something that Gopal does, and but for membrane proteins. And I won't go through it again. This is a somewhat old paper, but we could see that these things, doing Raman spectroscopy right around 1710. This is the red curve you're looking at. 1710, the Raman spectrum showed a peak, which showed this is antiparallel beta sheet. Okay, so that I said one of the main questions that we had, that these are antiparallel beta sheets. So I'm one by one testing the sort of criteria that I threw at the community and see. The next thing would be is it a specific, does it have a specific orientation in the membrane? So you know, if it's an ion channel. Yeah, sorry. Go ahead.
Previous slide, sorry to stop.
Next. Yeah.
So this is very interesting technique, but we know that whenever we expose these plasmonic metal nanoparticle with a laser, which is a strong light, it produces the hot electrons, it produces the electric field, and it also produces lots of heat. So is it not possible that the protein, which is directly sitting onto the surface of these plasmonic silver, may change their structure because of all of these, the electric field as well as the thermal heat that you produce on the surface?
Very good question. So fortunately, so I'll step back. So this is spectroscopy. So first, you don't need to put too much power, light power in it. But even then, it is one of the slightly insensitive spectroscopies, Raman spectroscopy, though it is actually helped by the SIRS effect. So you go with very little power, but even then we don't get any signal. So you have to build something which is called a confocal Raman microscope, which spreads out the power in a line and then does multiplex detection so that you can add up all that information and finally get some signal. So there is, yeah, the question is very valid, but we went to very low powers per unit area, and that is all. So, okay. Raman is a good indicator of conformational change also. So if you're hitting too much, you will see it in Raman. The spectrum will start changing. Okay, so then how do we know that it has a particular orientation? What do I mean by orientation? So as I said, it's ultimately a strand, even if it folds. There's an N side and there is a C terminal. There is the two terminals. Okay, so we put a fluorophore at. So by orientation, I mean the N terminus has to be at one position that all the N termini should be roughly at the same position, and the C termini with respect to the N termini cannot be in all orientation in the membrane. Then it doesn't have any particular orientation. Say the N is up, the C should always be down, or the C is up, N is down, or side, whatever, but the relative orientation should be maintained. Then only I know that it is taking a particular orientation. How do I test that? We again built this slightly new technique of these membrane things. There is a silver nanoparticle, and here we are not doing SIRS, not doing Raman spectroscopy, we're doing fluorescence spectroscopy, and fluorescence at some distance is quenched by the metal nanoparticle. And you can look at the quenching of the N terminal, quenching at the C terminal, and you can get some distance parameters, not exactly distance, but a relative distance. The relative distance showed that you have N terminal that is further out from the nanoparticle, C terminal that's closer, and this doesn't get averaged out. So they are not taking all kinds of orientation. So in bulk, it's there is a clear orientation. So that shows that also is that checks out. Okay. Then we are looking at placement. Where is the peptide in the thing? And this is absolutely new data. And we have these lipid molecules, and this is not something that we have published yet. We are still working with it. But the idea is that put radicals which can quench. Okay, this is something that Ranjan Das used to work with, these radicals. So radicals at different places, and the fluorescent particle that is there on top of the chain will sense these radicals in a process called triplet state relaxation. So that's a technical thing, but you can sense triplet state relaxation. So by putting the radical in different preparations in different places, you can sense where the, let's say, the N terminal is. And if you look at the N terminal, I'll skip through all the things. Just look at the right side. You see, this is sort of coming from this. This is the head group. This is 5. So if you count down the carbons from the head group down towards the middle of the bilayer, 5, 10, 16 goes to the absolute middle of the bilayer. And you see there is a clear pattern of location. So it's not randomly distributed all over, which also again tends to suggest that there is a clear structure here. It's not a random structure. Okay, so that also checks out. The last part is stoichiometry. How many do we have? This is the hardest part. We are not again quite there yet, the last two parts, but we had to develop our own idea of an existing technique, which is called single molecule photobleaching, where you count molecules one by one as they bleach. So you have a collection that are too close together to resolve. But if there are, let's say, 2 there, one turns off. It's like lights. The fluorescent reporters are lights. One turns off, then you see a step down. Another turns off, another step down, it goes to the background. So it shows each single molecule. You look at molecule by molecule. One single molecule got photobleached, this is the signal. Second one got photobleached, this is the signal. And you see there are only 2 steps and it became background. There is 1 step, there is 2 steps, there is 3 steps. So by looking at a collection, an oligomer, you can ask how many steps does it have till bleaching, and those many molecules it has. So that's the way you count. We are hoping to get a single number. Unfortunately, we do get a distribution. So the oligomers are not very well defined the way they are. Sometimes they're dimers, trimers, and tetramers, but at least they're not all over the place. Okay, so it peaks around trimers.
So I can ask a question.
Yeah.
This is Ulas here.
Yeah, Ulas.
So really, what determines this? What determines the stoichiometry of whether it is a tetramer or a trimer? I mean, because that was not clear to me.
Yeah. So if you look at the porin structure that nature makes, there are strands, many strands. That's a 6 to 24
No, but
strands.
in this particular case, I'm talking about
this particular case.
I'm saying in your particular. Yeah.
Yeah. So
what is
if the oligomer is a collection, that collection goes and sits in the membrane. Each monomer is carrying one fluorescent reporter. And I'm asking the stuff that is sitting in the membrane, which looks like a single spot to me because I cannot resolve it. It is sub-nanometer or nanometer scale thing. It looks like a bright light in my microscope. I can look at single molecules of light, but I cannot resolve them as single molecules. The brightness, sensitivity is there, resolution is not. But then suddenly these three molecules, the intensity drops a little, but doesn't drop all the way to the background. So, and then it drops again, and now it's gone to the background. So I have counted 2 steps, so that, and there is nothing further that happens. So I knew by sort of back calculation that that little ball of light I was looking at consisted of 2 molecules. It was a dimer. So that's how I look at it. And ultimately, if I think of the porin, each of the molecules actually is folded, so it can be 2 strands. And I think we need a minimum of 6, so it better have a at least a trimer. We do see dimers attached, but they may be not everything makes a porin. But we see trimers and tetramers.
Okay. The reason
so I have run out of my time. Sorry.
Okay. We will discuss this later.
No. I think Sudipta, you still have 15 more minutes, so you can go on.
Being generous, people will complain against.
No, it's okay. It's okay. It's all Zoom, part of the Zoom.
I will not take more than 10 minutes, and I'll go quickly to the 4th question. So forget all of that. So what we learned there is that there is a porin-like structure possible. We haven't proven it, but as of last year, I think all of the stuff that we are looking for checked out. Now it's really getting the full structure, which is very hard, but hopefully solid-state NMR will help us. But we know that it is a consistent hypothesis, which itself is much better than where you were 5-6 years ago. Now come to completely different things. Okay, small amphipathic molecules. Okay, they can, like cholesterol, they can also, there are other molecules called neurotransmitters. They can interact with the membrane, and we don't know what they do. So I'm saying an unknown window. The previous one was an intruder. It was toxic, we know, but this is, I don't know what it does. Nobody does. Now, where these small molecules, why am I talking about these small molecules? They're extremely important. Okay, so anytime anybody is taking an antidepressant, this is the molecule which you are trying to regulate. It's called a serotonin. It has a transporter through which it can go into the cell, and it has a receptor through which the neuron senses there is serotonin in the environment. So it is the messenger molecule, extremely important. Your learning, memory, especially in TIFR, all of us depend on those. Learning, memory, mood, your sleep cycle, etc. Many things depend on serotonin. Okay, and there are receptors and there are transporters. So it's a water-soluble molecule, and as of now, as we understand it, all it needs to do is be in water and go and hit the transporter or go and hit the receptor. No need to interact with plasma membrane at all, because those receptors and transporters are sitting in the plasma. But what people found out, about 8 years ago, that serotonin binds to lipid membranes very strongly, which was something that, who ordered that? We didn't need it. We didn't need this property. And yet it has, and it binds to, if you leave serotonin in water, it does partition between water and this thing at concentrations of, I think, a millimolar or so. It is 1,000 to 1. 1,000 molecules will go to membrane, 1 molecule will stay in solution. Though the solubility is high, you can go molar quantities in solution, 1 molar, 2 molar quantities. So it is surprising that it goes into the membrane so much, and nobody knows what it does. But if it is going to that extent, we thought about 2 years ago that it must be doing something, and that's where all this started. How to probe that? We know that it is going and sitting in the membrane. That is easier to check. We have some fluorescent techniques for doing that. But what is it doing there? Well, we decided, okay, it may be changing the stiffness of the membrane. And it's not something that fell out of the sky. The cholesterol that you love and hate, is something that changes. It's known to change the stiffness of the membranes, by which it regulates lots of processes. As I said, that your neurons won't be functioning if they didn't have enough cholesterol. So it had to maintain the right rigidity, not too much, not too little. So we went and started poking the membranes. And these membranes, these are artificial membranes you can put on a dish. You can poke it with an AFM probe, and you can go ask when this AFM probe breaks through the membrane and hits the bottom. The bottom is a mica sheet or something is hard. So when it goes and pokes through, there will be a little, so this is deflection of the AFM tip as it senses the force versus the translation. And you see this signature, you can measure the force at which the membrane gives. You're making a hole in the membrane, the AFM tip can go in. So I said, okay, that's a good way to measure some measure of stiffness. Does serotonin change that? And it did, hugely. So what we found out is that without serotonin, a particular type of membrane, which is a very artificial membrane, has 2.5 piconewton of, nanonewton of force before it breaks. And that with 5 millimolar of serotonin becomes about a nanometer. Factor of 2 change in the force, and 2.5 to 1.14 if you want. So it seems like it's making membrane more disordered. Okay, as of now, Ankur, my student Ankur, will raise his hand and says, you don't understand what you say when you may say something like disorder. I agree, but I'll use the term loosely. Something that is a collection of these lipid molecules is easier to make a whole. That means probably it's becoming more disordered. At least that's what, that's one way of talking about it. So again, we went to solid state enema, this time to Daniel Huster, his lab in Leipzig. We asked, are we reading it right? Can you do an experiment? Okay. Of course, in fluorescence, you can do an experiment in a single, or AFM, you can do it in a single layer. In the, NMR, you have to supply a lot more sample, but that's okay. He did something called magic angle spinning that Madhu also does. And he can look at atom by atom and its disorder. Disorder comes in, even in a membrane, things are slightly dynamic. So things are rotating about, and that you can see in the NMR spectrum, and you can deuterate things and all. And he gave us this graph, which is very interesting. You look at the level of disorder, this is called an order parameter. Okay, order parameter has different connotations in different parts of physics, but there is an order parameter in the sense how disordered things are. Order parameter reduces, it's more disordered. So this is no serotonin, this is 10%, this is 25% serotonin, and this is at 25 degrees. You look at one lipid chain versus the other lipid chain, you get different, but in every case, you see a reduction. And he can actually talk about where that reduction is. So he has a molecular level picture. So our ability or our guess that by AFM, it is actually becoming disordered, turns out to be true in this case at least. Okay, that opens up huge possibilities. If membrane order can be changed by serotonin, that nobody knew, everybody thought serotonin is just doing some signaling, then it's a very big news, because membrane order changes a lot of things. Ability of another protein to hit the membrane, to insert in the membrane. For example, our own oligomers. Okay, in this case, it's IAPP, is slightly related to amyloid beta. Their ability to attach to the membrane completely depends on membrane order. What you see in the right here are disordered phases, and O is ordered phase, and we know that it goes into the ordered phases because we have constructed something called an AFM confocal combine. The green part are the disordered phases here, and green is where the IAPP oligomer is going, and this is AFM image, this is confocal image, and they are going 7:1 into the disordered phase. That means your ability to be attacked by the oligomers, your vulnerability of your cells to be attacked by the oligomer increases if you have serotonin around. Serotonin membrane is changing. And this actually also affects cells. It's not just something in the lab. And we did experiments. We do a lot of experiments with neurons, which we grow in the lab. And you can see in fluorescence images that you have these are rat neurons. You have the brightness actually comes from the oligomers carrying a fluorescent reporter. It becomes brighter in presence of serotonin. And it becomes about 25% brighter in average. That itself is quite significant. And which means that, yes, even the cell membranes are becoming vulnerable. And I think I will stop in the next 2 slides. Oops. Here, and it shows that forget about something else attaching. Process of endocytosis. If the cell membrane becomes less stiff, I showed you that little cartoon where the membrane has to bend, something that Devashish works on. Ultimately, it pinches off. And that bendability is something that is improved, or this is helped by serotonin. Then we hypothesize it'll be easier for the cell to do endocytosis. And we've got a hint of that. There's endocytosis of something called transferrin. I won't get into this. It is something that the cell is doing all the time. This is how the cell eats iron, okay, by endocytosis of transferrin receptors. That goes up, the rate, basal rate goes up by about 35% in presence of serotonin, okay, which doesn't quite prove that we have a serotonin-dependent effect in real biology, but shows that the possibility that was spawned by the observation that serotonin interacts with membranes can actually have biological effects. And it's not just serotonin. Here's my last slide. You look at all the things that Ankur has been able to do now, the force measurement, and whether the black is all without the neurotransmitters. They're all the class of neurotransmitters that serotonin belongs to. They're called monoamines, very important neurotransmitters. Here is norepinephrine. This is how much difference 0.5 millimolar of norepinephrine makes.
Hello?
Hello?
Is it frozen?
Yeah, I thought I lost my connection.
No,
Can you
looks
hear
like-
me? Can you hear me?
I can hear you.
Everybody can hear me?
I can also hear you.
Okay. Sudipta? Sudipta, we lost you. Okay, we lost him completely. Wait, I'll request all the audience to wait for a second. Hello. We lost you. We lost you. Okay.
If I don't come back, I'll just then I will think on your microphone. I'll just completely stuck.
You are rebooting. Okay. We are waiting. Yeah. Okay. Sorry, guys. He's having some computer issues. His computer just restarted. Hello. Can everybody hear me?
Yes, JD. Yes, we
Yeah.
hear.
Okay. Yes. So just we are waiting for next few minutes. Let him finish, Ullas. We can have the discussion after that. Is that okay, Ullas?
Yes, boss.
Yes. Boss. All of this is getting recorded, Ullas.
No comments.
Okay.
While we are waiting, can
Yes,
I
Allah.
ask a question to Ullas or any of the biologists?
Yes, please.
So he showed that norepinephrine
Yes.
a very strong,
Effect on the
effect, right?
on the membrane.
Exocytosis. So do you think
Yes.
among all these neurotransmitters, the norepinephrine is a real, oh, sorry, Sudipta is back, so I should shut up.
No, so you should ask him this question. We can get back to yeah. Sudipta is saying something. Wait, Sudipta is saying something. Hi, Sudipta. Yes. Can you unmute yourself, Sudipta? Can you unmute on the Zoom? Yes.
Can you hear me now?
Yes, perfect.
Apologies, and I'll finish in a minute. Enough.
We were at norepinephrine. Effect.
That was the last slide, and it sent something and quit on me. So
Alok already had a question. Without you finishing, so Alok has a question. So
okay, good. Let me just share the screen and get to it. So I'll just stop in this thing. Okay, let me just go where I was. There. Okay. So Alok, just give me a minute so that I can finish it, and so that then I won't take any more time of other people who may want to leave. Just hold on to your question. So, so this is my final slide. Some answers we have in this new journey. Serotonin and many other neurotransmitters interact strongly with the membrane. They change the mechanical properties of the membrane, and this changes the propensity of other molecules, other proteins to attach to the membrane, potentially playing a role in amyloid diseases, where some connections with serotonin and other neurotransmitters are known, clinically observed. This also changes the ability to exocytose or endocytosis of transferrin. And the last bit I was showing with norepinephrine is that this effect is very species specific. Not all monoamines do it. It's not like a generic chemical effect. So nature has played with some things, and maybe nature has evolved some molecules, some of which can do it, some of which cannot, some of which probably do it the opposite way, we do not know. So ultimate message is monoamine neurotransmitters, some of them can modulate cells using a receptor-independent pathway. The receptor-dependent pathway everybody knows about. Okay, it's a receptor-independent pathway. Let me do the honors to the people who actually have done this work, and all of us, I know why we actually are in TIFR. It's for the fact that TIFR offers such a great bunch of people. Okay, you absolutely get the best students, and I'm so thankful to them. There's Vicky, Ankur, Anup, Devshankar, Anirban, and Arpan. You can see, of course, they are the current students. And what I talked about, staff talked about, there are lots of others. Madhuri and Aditi are two of the current members you don't see in this picture. This was taken in February, and on top of the rooftop restaurant when the COVID was slightly down, we just felt like celebrating something. And of course, the second big reason why you join TIFR, you also get great colleagues. Of course, my collaborators come from all over the world, but you'll see quite a few from TIFR. Madhu and Ravi are parts of the work that I have talked about. And of course, Daniel and Gilbert are very good friends who are not TIFR people, but are parts of the work I talked about. But I'm absolutely delighted that there are lots of other people that I'm working with, Kanchan, Aditi, Debashis, who are all parts of TIFR. And Peri, I used to work with a long time ago. We had a very important paper together. And there are other people who have contributed. So DAE and DBT, of course, have supported the work. And thank you very much. I'm sorry I ran over time and then my computer quit. Thank you.
Thank you very much, Sudipta. So I would like all of you to please unmute yourself and just have a big round of applause for Sudipta. A very nice talk. This is something that we miss in these Zoom sessions is the personal sort of applause that we have reserved for the speaker. So
I missed a lunch and I missed a coffee. Paid for by somebody else.
Yeah, sure. We will try to do something in future. I request all the participants here to now ask questions freely. Just unmute yourself and fire.
Eddie, I have a question.
Yes, please. Yes,
Hi.
please.
Sudipta, very nice talk, and
thank you.
Yeah, comprehensive experimental data to convincingly say that A-beta basically gets attached to membrane, not only gets attached to membrane, but might be forming porine-like structure. But then what is the next step? So let's say if you need to get the structure of, let's say, porine forming of oligomer, is it possible one can use, let's say, covalently linked multimer of A beta, okay, to use so that it might form, because right now you are using unlinked aggregate oligomers, which are attaching to membrane. Is it possible to use, let's say, covalently linked tetramer because it's antiparallel beta strand, right? So is it possible to use, let's say, covalently linked tetramer, which might have higher propensity and also form porine-like structure on the membrane? Can you comment?
Some people are trying that, but you know, Koti, you and I know that if I give you 4 molecules, they can be covalently linked in many different ways. So unless you have an idea about the structure to start with, you won't know how to covalently link them. But there's absolutely the way to go. Once you know the structure, to artificially make it and study all the properties, and who knows, I mean, an antibody to that molecule itself may solve A beta, if all the stars are aligned. But the point is you need to know the structure. If you know the structure, you may even come up with a small molecule. So it will give an enormous window of opportunity to, let's say, pharmaceutical companies to do something about it. But you have to know the structure.
Okay. Okay, Ullas, you raised your hand, long time. Yeah.
Yes, Sudipta. So, very different question, but something related. So, there should be a thermodynamic barrier for proteins during evolution and the extent to which amphipathic regions should be incorporated in a protein. So, if you look at related proteins across evolution, or when there is new exonization that happens, which we know all the time happens, is there certain allowed and disallowed composition of peptides based on which your genome sequences can be now predicted to form or not form peptide sequences or protein sequences? What are the thermodynamic limits where when exonization is happening all the time, new domains are being evolved, you should have a balance between how much of amphipathic nature should be allowed and how much of it should be disallowed, both for that particular protein, but also in general overall mass of the proteins that are present in the cell. So, I mean, is it at all possible to look at these things from a very global perspective? Your thoughts?
Yeah. So it's a very good question. So one of the things that is studied now very well is the foldability. As I said, I briefly mentioned, proteins have not evolved just for the function that they do, just for optimizing that, but also for optimizing foldability. And these concepts, the initial concepts came from a solid-state physicist, Peter Woliness, and he introduced the concept of frustration. So the free energy funnel needs to be reasonable. It will be rugged, but reasonably downward so that you don't get stuck. Otherwise, if you have random places, hydrophobic things, and you have multiple possibilities by which the protein can minimize its energy, then it will not get anywhere. So you need to have this foldability built into the sequence. And I think the only way nature has learned it is by making mistakes, and probably is always making mistakes. You know how the SARS-CoV-2 is evolving, making mistakes, but making sure ultimately those things are surviving which are foldable. Because the uniqueness is also important. You can't have just a strand, a protein which can fold in 20 different ways. It's not going to do, as you and I know. So yes, foldability is something that, and the ruggedness of the energy landscape while having a downwards funnel-like thing is very important. And there's something that I think only got appreciated in the last 2 decades when solid-state physicists came into it and talked about frustration, that foldability must have been one of the big things that nature had to take. There could be protein designs which are much higher in, let's say, ability to, say, carbonic anhydrase kind of thing, and a huge ability to do enzymatic catalysis. But if they're not very well foldable, then nature can't have it.
Yeah, so question really that I was trying to drive at is, as evolution has proceeded, if there is more selection towards protein sequences or stretches which are more foldable or which can we have a lot of amphipathic stretches, then you are paying the cost by possibly also allowing them to aggregate and form amyloid-like structures or form random aggregates that you don't want. So is that what you see? Again, I'm just asking from an evolutionary perspective. If you look at related proteins, related families of proteins, if you look at phylogenies, is that something that jumps out?
So that's an interesting question from the evolutionary point of view. See the kind of parallel beta sheets that these proteins which have hydrophobic patches exposed, they can easily make. And it seems to be a kind of a generic phase of a protein. So some people are reverting that, saying that instead of thinking it as a pathological thing, think of it, these are probably the earliest kinds of structures that protein could form, because you didn't have all the sophistication of the design that came through billions of years, but it is some generic design and possibly did something, and we have remnants of that still existing in silk fibroins and things like that in structural proteins, where this is used. And perhaps this is sort of the earliest principle, this parallel beta sheet. It's called a cross beta architecture by which proteins got organized and made larger structures. Yes, but nowadays, obviously proteins are much more sophisticated. And anytime you try to boil a protein, there's a tendency it will go into this. But that's like the basic tendency. It's like the basic instinct kind of thing.
Okay.
Okay.
Thanks.
Okay. Thank you, Ullas. I request Alok Ray, please unmute yourself.
Yes, thanks. Sudipta, I have this question. You showed towards the end that this neurotransmitter norepinephrine was far more effective in changing the mechanical properties of the membrane than other similar neurotransmitters like perhaps serotonin or something. Now, if if this is a desirable property to change the mechanical property of membrane, then why wouldn't nature favor, nature and evolution favor norepinephrine over serotonin as the preferred neurotransmitter?
Very good question. So obviously you have like 200 neurotransmitters in your brain.
Not desirable, then it's the opposite kind. Why doesn't it weed it out?
So yeah, you have about 200 neurotransmitters in your brain, and obviously a few of them will have this effect. The answer to that would be that it's not in general desirable effect. The nature may be using it in some kinds of cells. So the way your brain is organized is that different neurons typically carry different messenger molecules. Some neurons by design may be such, involved in that kind of neuronal activity, which a longer lasting change may be desirable. For some other neurons or for it in a different circuit in the brain, it may be a non-desirable activity. This is something which is lengthens the time scale, makes it more diffuse when signaling is happening. So there is a diffuse memory of signaling going on that serotonin is doing and norepinephrine is doing a lot more. And maybe for few neurotransmitters, for few types of neurons involved in few types of functions in the brain, that will be required. So the answer is that horses for courses.
Okay.
Thank you, all.
Okay.
Thanks.
Okay. Okay. So Devashish has a question. Devashish.
Yes, Jyotishman. So, Sudipta, I have a question. This is regarding another protein like alpha-synuclein. So, alpha-synuclein also, aggregates. It causes Parkinson's and everything, and it interacts the membrane in the same way say beta, does, right? So now, when it is monomer, it also interacts with the membrane, and not just interacts. I think for the functionality of alpha-synuclein, this membrane interaction plays a key role, and people have shown it. Now, what I'm trying to understand, like, what is your thought regarding, say, interaction of monomeric alpha-synuclein with the membrane and say oligomeric alpha-synuclein with the membrane? What do you think? And with a like a chemical difference between these two? I'm more interested to understand from the protein perspective, not from the lipid perspective, because lipid can vary and all these things.
Okay, so the big difference between amyloid beta and alpha-synuclein is the size.
Okay.
So amyloid beta would not be able to make any reasonably complex structure in the membrane or outside without first becoming an oligomer. But alpha-synuclein is much bigger, and therefore a single alpha-synuclein molecule can make some kind of a structure in the membrane. And I know it is important for synaptic activity, people have shown.
Yeah.
Now, such a big molecule which can do something by itself, but it is actually unstructured mostly, even though the size is there, in principle, which probably is one of those frustrated proteins that cannot fold properly. But when it goes to the membrane, it probably takes some kind of a structure. Why it then aggregates? Why it aggregates thermodynamically is easy to understand. There's lots of hydrophobic residues exposed. But in that big oligomer, what it is doing in the membrane is a question similar to a beta question, but probably harder to answer. But it's known to be toxic. So whether it's a similar mechanism, whether it's a different mechanism, that's a good question. We do not know. What we did the last few years is that as a counterfoil of A beta, we took another molecule called IAPP, which is the causative agent of type 2 diabetes, because they have similar length. IAPP is 37 residues, A beta is 40 to 42. And between them, these two, we see lots of differences. Okay, there is not like a generic principle of something. So we have not dared touch alpha-synuclein with such structural detail, but someday we will. But yeah, it will be much more complicated because the size makes it a very different model. So I guess the short answer you guessed, I don't know.
Okay.
Thank you.
Thanks.
Thank you, Devashish.
Yeah.
Sudipta, I have a question with regards to the membrane effect that you found for the neurotransmitters. So you've shown this, and I've heard this multiple times from presentations that Ankur has also given across different meetings. The fact that when we talk about membrane disorder, it really depends on what kind of measurement you are doing, right? You will agree that most of these measurements, when we look at membrane disorder, we are doing an atomic force microscopy image or an imaging modality which can measure the length of these lipids, in the sense that who is standing, the ones that are ordered are standing taller than the ones that are disordered, that are bent, and they don't reach that height, that complete height. They don't have the complete length transcribed as a translipid chain. So the question then, to compare it with NMR, which is a very, very local structural probe that only talks about what the protons are doing. That's what you're measuring in this case, the proton mobilities. And that is an order parameter on the local sort of length scale of things. So I wouldn't be surprised if not only serotonin, but other kinds of molecules also perturb that local ordering that you measure from NMR. So that may not be the sole perspective when we compare it to the AFM height images, for example. And there could be other ways of looking at the problem, like fluorescence tagging, as you have shown. So can you comment on this, that when we are looking into these mechanical properties, how does these spectroscopies actually end up giving you a complete knowledge of what is going on?
So you have hit upon something that we are, of course, worrying right now. Very interesting question. What you're saying is that the AFM indentation force, that is easier to make a pore, need not really correspond to anything that you measure by NMR, because that's a molecular level thing.
Right.
And this is kind of it's the whole body. It's like looking at individual bricks and seeing why did the house fall down. The house may have fallen down by different reasons.
Right.
Precisely. So the point is, understanding membranes is extremely important for biology, that different processes which may be affected by different things. Some processes may be affected by what individual local things are doing. Diffusion in the membrane plane, for example, may be dependent, may correlate very well with the NMR.
Right.
But think about pore formation, think about endocytosis. I would say the AFM is kind of an aggregate effect. Which part is giving proper force, proper resistance, ultimately what is giving way. It need not really correlate, you're absolutely right, with the NMR order parameter. But if I have to correlate it with a biological phenomenon such as pore formation, which is really what I'm after, then that sounds more appropriate than something else. So we are after that. But this whole thing, as you know, have given rise to this whole single notion of membrane order seems nonsense. Okay, in a sense, you are saying, okay, you have given me two different things, and there's a complex object. There's nothing like a single notion of a membrane order. So we have to relearn the things, and many people, chemical physicists and biologists, use one reporter of membrane order to predict everything about the membrane. And the main point is that's also complete nonsense.
Not
you
right.
cannot just
Not
measure one
right.
and say that property will get affected. So there has to be correlation between the property in biology you're looking at and which measurement are you looking at. And yes, you are right, NMR and this thing need not always correlate. In fact, that's what we are trying to find out with many more membranes and many more measurements now.
Right. Okay. Thank you, Sudipta. Are there any other questions? Well, if there are none, then I would like to thank Professor Sudipta Maiti once again for a wonderful colloquium.
Thanks,
and
students.
Very nice talk.
And yeah, and I also want to thank everyone for joining, and just want to remind the 20-odd people who are still here with us that don't forget to join in for the next colloquium session, which is going to be about results from the Event Horizon Telescope, the EHT, that has recently reported some very exciting results about light and magnetic fields from a black hole, from the edge of space and time in a black hole. So Professor Ivan Marti Vidal from University of Valencia is going to describe a multi-country effort to understand M87, which has a black hole at the center of the galaxy. So so thank you all for joining in, and I wish you stay safe amongst this pandemic time again. Thank you, Sudipta. Thank you. I'll close.